A slow-wave potential is a rhythmic electrophysiological event in the gastrointestinal tract. The normal conduction of slow waves is one of the key regulators of gastrointestinal motility. Slow waves are generated and propagated by a class of pacemaker cells called the interstitial cells of Cajal, which also act as intermediates between nerves and smooth muscle cells. Slow waves generated in interstitial cells of Cajal spread to the surrounding smooth muscle cells and control motility. Description: in physiology, the slow wave threshold is the potential which must be reached before a slow wave can be propagated in smooth muscle. Slow waves cause no smooth muscle contraction. When the amplitude of slow waves in smooth muscle cells reaches a certain threshold, the slow wave threshold, a particular membrane ion channel, L-type Ca2+ channels, are activated, resulting in calcium influx and initiation of motility. Slow waves are generated at unique intrinsic frequencies by the interstitial cells of Cajal, even within the same organ. Entrainment of these different intrinsic frequencies through electrical coupling allows these unique intrinsic frequencies to occur at a single frequency within the stomach and segments of the small intestine. Electron microscopic and decoupling studies to date have confirmed gap junctions as the major coupling mechanisms between interstitial cells of Cajal. Coupling between ICC and smooth muscle cells is uncertain. Gap junctions have been demonstrated in rare circumstances as one coupling mechanism between ICC and smooth muscle cells. Another potential coupling mechanism is the peg-in-socket theory, which demonstrates that the membranes of smooth muscle cells have the ability either form physical narrow sockets or pegs to lock onto other smooth muscle cells and/or interstitial cells of Cajal. Types: gastric slow waves occur at around 3 cycles per minute in humans and exhibit significant variances in both amplitudes and propagation velocities in the stomach due to the existence of a gradient of resting membrane potential gradient, interstitial cells of Cajal distributions, and gastric wall thickness. Gastric slow waves frequency, propagation velocity, and amplitude demonstrate significant interspecies differences. Extracellular bioelectrical recording studies have demonstrated that gastric slow waves originate from a pacemaker region located on the greater curvature of the stomach. Human gastric slow waves propagate slower in the corpus than in the pacemaker region and antrum of the stomach. Up to 4 simultaneous low-wave wavefronts can occur in the human stomach. Intestinal slow waves occur at around 12 cycles per minute in the duodenum and decreases in frequency towards the colon. Entrainment of intestinal slow waves forms frequency plateaus in a piecewise manner along the intestine. Similar to the stomach, intestinal slow waves frequency, propagation velocity, and amplitude also demonstrate significant interspecies differences. In uterine smooth muscle, slow waves have not been consistently observed. Uterine muscle seems to generate action potential spontaneously. In gastrointestinal smooth muscle, the slow wave threshold can be altered by input from endogenous and exogenous innervation, as well as excitatory, acetylcholine and substance P, and inhibitory, vasoactive intestinal peptide and nitric oxide compounds. References: Textbook of Medical Physiology, Guyton and al., 12th edition.